Hello friends, again welcome to my channel. Today we are going to discuss about the properties of ideal bioreactor, which is a point included within the industrial microbiology. Now the bioreactor or the fermenter, it is any type of the manufactured machine or the system which is responsible to support the biologically active environment. Means the bioreactor, it is a specific type of the vessel which is responsible to provide all essential condition, either these are the physical or chemical conditions which are required for the growth of the microorganism and for getting the specific product from that microorganism. All types of the bioreactors, these are deals with the heterogeneous system. That means it involved two or more phases, that is the solid, liquid and gases. All the optimal condition for the fermentations, these are requiring the efficient transfer of mass, heat and momentum from one phase to the other. The chemical engineering principles, these are utilized for the design and operation of bioreactor. Now we are switch over to the characteristics of ideal bioreactor. So the ideal bioreactor or the vessel, it should be capable of being operated aseptically. Means the aseptic condition, it must be maintained within the bioreactor for the purpose to avoid any type of the contamination, as within this process or the any fermentation process we are utilizing the pure cultures, so there are no any chances for the contamination within that vessel. Then the second important point, it should must be provide the adequate aeration and the agitation provided to meet the metabolic requirements of the microorganisms. Now, through these both processes, that is the aeration, it will require the adequate supply of the oxygen to the microorganism for the aerobic fermentation process, or for the whenever there is the requirement of oxygen for the microorganisms. Agitation, it is responsible for the proper mixing of the fermentation medium to transfer the all of the nutrients within the fermentation medium to each and every cells which are growing within that fermentation medium. However, the mixing should not cause the damage to the organisms. The power consumption should be as low as possible, which will responsible to reduce the cost. A system of temperature control should be provided. Now, within the different types of the fermentation process, there may be the changes within the temperature. Either the temperature may be cooled or the rise due to the microbial growth. So, within that conditions, majorly it is responsible to maintain the constant temperature. A system of pH control, it is also important, because we know that these are the important two physical parameters which are responsible for affecting the growth of microorganism. Whenever there is alteration within either the pH or the temperature, they are responsible for affecting the growth rate of the microorganism. Then the sampling facility should be provided. Now the sampling facilities, it means whenever within the continuous fermentation process we are up to the after the specific time period, we are going to operate, or we are going to transfer the specific term amount of the fresh media within the fermentation process or within the fermented. So such type of the sampling facilities should be provided. Then the evaporation losses from the fermented should not be excessive. The vessel should be designed to require the minimal use of labor in operation, easy to operate, harvesting, cleaning and the maintenance. Ideally, the vessel should be suitable for a range of processes, but this may be restricted because of containment regulation. The vessel should be constructed to ensure the smooth internal surfaces, using the welds instead of the flange joint whenever possible, because they are may be responsible for creative problems during the cleaning process. The vessel should be similar geometry to both smaller and larger vessels in the pilot plant or the plant to the facilitate the scale up process. The cheapest material which enable the satisfactory results to be achieved should be used. There should be the adequate service provisions for the individual plants. Now the bioreactor should provide the following conditions, that is the agitation. Now the agitation, which is important for the mixing of cells and the medium, that is the proper transfer of the nutrients to each and every cell. Aeration, that is for the proper oxygen supply. Regulation of factors like temperature, pH, pressure, aeration, nutrient feeding, liquid level. The sterilization, proper sterilization, it is important to avoid any type of the contamination. Then the maintenance of sterility, no any type of the leakage which may be cause the contamination. The withdrawal of cells or the medium for continuous fermenter. During the continuous fermentation, majorly there is the continuously addition of the fermentation broth and the removal of the product. So for that purpose it is important. The modern fermenters are usually integrated with the computers for efficient process monitoring and the data acquisition. Generally, the 20 to 25% of fermenter volume is left unfilled with the medium as the headspace to allow the splashing, foaming, and the aeration. The fermenter design varies greatly depending on the type and the fermentation for which it is used. Means for the various types of the application, the various fermenter designs these are available to carry out such type of the fermentation processes. The bioreactors are so designed that they provide the best possible growth and the biosynthesis for industrially important cultures and allow ease of manipulation for the all